Animals respond to stimuli in their environment. In fact, the definition of behavior is a response of an organism to a stimulus. This little video demonstrates clearly a stimulus-response relationship, an animal responding to a stimulus. This is a bee. She's in a halter where she can't move around. Her eyes have been blackened over so she can't see the instrument that's being applied to her antennae. She's being presented with a droplet of sugar solution, sucrose solution. And when she touches it with her antennae, she sticks out her proboscis, and that is her response to the stimulus. The stimulus was the sugar solution. She won't extend her proboscis unless the sugar solution exceeds a certain threshold. So she demonstrates a threshold of response for that sugar stimulus. The neurons that are attached to the receptors on the antenna that perceive the sucrose solution also have a threshold of response. This top figure here shows a neuron and the different parts of it. Out here we have dendrites. These are inputs. They might come from the sugar receptors or some other kinds of receptors coming in, or maybe they're attached to other neurons that are attached to receptors, and they're just part of a chain of information flowing through the neurons. But the dendrites then pick up signals and send electrical signals down to the soma of the cell, the central part of the cell. The signals come into the soma, and then the soma will either fire an action potential or it won't fire an action potential. If it fires an action potential, then the action potential travels down the axon to the axon terminal, and these axon terminals may be attached to the dendrites of other neurons, which then take the signal and propagate it. Maybe it goes into the central nervous system and gets processed there, or maybe it goes to motor neurons, which then stimulate a muscle that then contracts. The key here, though, is for the signal strength that comes into the cell body, the soma, to be sufficiently high that it can trigger this action potential. If you look at the action potential has a resting potential. And this is the potential difference in electrical charges between the inside of the neuron and the outside of the neuron. In the case of a honeybee, it would be in the hemolymph that's outside of the neuron. If the charge gets high enough to exceed the threshold of the excitation threshold, if it gets above that, then it will propagate the signal down the axon by opening up ion channels and letting those ions, particularly the potassium ions, flow out of the cell and into the hemolymph that surrounds it. But you can see here that there's a threshold of excitation. And if it doesn't get excited, if the inputs into the cell don't exceed this threshold, it won't fire the full action potential. So how do they do that? How do they integrate this? Well, they can integrate these signals either temporally or spatially. They can undergo a process of summation. So for instance, on the left here, we have a neuron at the top, and it's getting stimulated to the dendrites. It's getting stimulation to this one here. And it comes in, and that stimulation is not enough to exceed that threshold of the cell, of the nerve cell. And then it gets another one, and it's still not big enough. So this neuron is not going to fire an action potential down its axon. It's not going to do it. But some of them will sum them. So the first one comes in, the first stimulus comes in, and it elevates the resting potential to here. And then the next one comes in. If it comes in at a very short time interval close to it, it might add it on top of it and get to here, and then it'll trigger the depolarization of the cell, and the action potential will propagate down the axon. But they can also integrate spatially. So this would be integrating over time. So if you're getting rapid succession of stimulants, then they build up and then it'll fire the cell. In this case, it would be spatial. So you could be having multiple inputs into the cell, like an input here and an input here. And if these two come in and come into and get transmitted into the soma, the cell body of the neuron, then it adds here, then it adds up, and then it fires. You fire the action potential. So this is the level of threshold response at the level of the neuron. But we think about this in the context of individuals. When we're looking at behavior, we kind of, what's going on at the neuronal level and all is just sort of absent from us. We don't think about it that much. But this is the case here where we have 4 individuals, and each of them has a different response threshold to some stimulus. The stimulus could be the sugar being touched to the antenna of the bee, and the response could be extending the proboscis, like we saw in the video at the beginning. In this case, we have 2 individuals that have thresholds of response that are higher than the actual stimulus level. They're not going to respond. And whereas here and here, their thresholds of response are below the stimulus level, so they will respond. And we can see this. We can measure different bees with sugar concentrations. We can increase the concentration that we present to their antenna. Some of them will fire at 3%. It'll trigger their response. Others, it'll take 10% or 30% before they'll respond. So they vary in their response thresholds to the sugar stimulus. We can also look at us. This is a cartoon I had drawn. Actually, I had it drawn from actual pictures that I took. Anyway, so we have a stimulus. It's dirty dishes in the kitchen. And they're piling up. And there's 2 people who live together here. There's this one and this one. And this one here, she has a low response threshold for dirty dishes. So she steps in and she washes them. When she washed them, she eliminated the stimulus altogether. And since the stimulus was eliminated altogether, this individual here had a higher response threshold. The dishes will never pile up high enough to exceed his response threshold to where he'll actually do the dishes. She will always respond, always remove the stimulus. He won't do it. So he'll do something different. This is the basis of division of labor. Individuals have different response thresholds to different stimuli. Some respond to it. When they respond to it, they actually reduce the stimulus by their activity. Her activity here reduced the stimulus from here to there. And when they reduce it, then that basically walls off others from doing that task. So then the other individuals encounter something else to do. So you get a natural division of labor, whether it's chores in your house or it's chores in the nest. Honeybees have a special problem. And we have receptors all over our skin and all over our tongue and whatnot. But honeybees are hard on the outside and soft in the middle. They have a hard exoskeleton, the shell. So they have to get receptors somehow through that hard shell to experience the environment around them so that they can get information. Those receptors come in different types. You have tactile receptors, which are touch receptors. There are chemosensory receptors of 2 types. There's the olfactory receptors, which can pick up odors in the air. And then there's the gustatory or taste receptors, which are also contact chemoreceptors that they use for taste. They have the gustatory receptors are located on the antennae, the mouth parts, and the tarsi of the feet. So you can imagine a bee stepping in the nectary of a flower, and then the concentration exceeds her threshold of response and her tongue comes out and she sucks up the nectar. Visual receptors, they're used for vision and to detect motion. Thermal receptors, they have receptors that can determine temperature. And water vapor receptors that can determine humidity. They have all this kind of information is coming into the central nervous system, into the central processing units of the brain from the exterior of the bee by receptors that are on the outside of their skin, their hard exoskeleton. This is an electron micrograph, a close-up of a honeybee antenna at the basal end of it near the head. And the top one shows contact receptors. You have all these little hair-like things all over that are receptors of things that the antenna touches. They get information through those. And then there's these branched hairs. The branched hairs are not receptors. Branched hairs are there to collect pollen. When they get into a flower, there's a lot of pollen in the flower. The pollen sticks to these branched plumose hairs, and then they go and they comb it out, and then they collect the pollen and put it in their pollen baskets and take it back to the nest. The types of sensilla are the types of receptors that they use. We call these receptors sensilla, or a single one is a sensillum. They're based, the different types are based on their shape. It's basically the morphology. But there's a type called a trichoid sensillum, and it's primarily used in tactile sensing. It can detect touch. So those setae that you saw on the antennae of the bee, each of those hairs is set up like this. It's a hair that sticks out, and then here's the neuron down here, the soma, the body of the neuron, and here's the dendrites here, and these are little branch dendrites, and then they detect movement by the deflection of the seta back and forth, and then they transduce that signal, firing it down to the cell body. And if that signal is strong enough, if maybe it's summed temporally, or it might be summed in some other way, then if it's high enough, it'll fire the signal down the axon. It will go back into the central nervous system to get processed in the brain, and then some sort of an action might take place. Another kind of touch receptor is the campaniform sensillum. And it's basically this is the integument, the cuticle on the outside of the bee. And you find these on the antennae also. And it has a cap. It's not a hair, it's a cap. And then these are the dendrites, and they're attached to the inside of the cap. And they can detect pressure. So any kind of pressure on the cap will cause some sort of disturbance to these dendrites that are attached to it, and can fire a signal back down to the neuron, the cell soma. Basiconic sensillum, these are for odor. They're basically shaped like a little hair or a little peg, and they're full of pores. And then inside of these pores, the dendrites come up into the seta, and then inside of these pores there are specific chemical receptors, and they may be general type chemical receptors, or they'll be maybe very specific odor receptors. That then they'll detect these odors and then fire the signal back down to the neuron cell body. Coeloconic sensillum. These are used for detecting odor, temperature, and humidity. They're located at the bottom of pits. So here's the cuticle here, and this is a pit in the cuticle. At the bottom of the pit is this little projection, and that is the sensillum that then can detect odor, temperature, or humidity. Looking closely at a honeybee antenna, we can see here's the trichoid sensillae again. These are the touch receptors. And this one here is a gustatory receptor. an odor receptor, and excuse me, not a gustatory, an odor receptor. And these are just plate-like objects on the surface. And inside of those, there's a lot of different pores and receptors for different potential chemical odors. Here's a siliconic syncyllum. It's a peg. These are most often gustatory. Now, the touch receptors, there's lots of different kinds of touch receptors and lots of different kinds of odor receptors, but there are far fewer different kinds of gustatory receptors. You find the gustatory receptors on the antennae, the mouth parts, and the feet, as I said. And they can also detect water vapor, but they're just a little peg with a little pore on the top of the peg to detect the odors. The eyes, the visual system. The honeybee eye is composed, each eye is composed of 6,900 ommatidia. Those are the individual simple eyes. The overall total eye is called a compound eye. And it has, as I said, 6,900 different ommatidia, which are individual eyes. This cross-section here shows it nicely, that each of these ommatidia in the eye, if you look at them in cross-section, they all funnel the light down here to where there's a sensor, a light sensor, which then transduces the signal back into the optic lobes. So you can imagine that a bee patrolling, walking around in the nest, encountering stimuli. And the stimuli that it encounters and the response threshold it has to those stimuli will result in it behaving in a certain way or not behaving in a certain way. So bees spend a lot of their time what we call patrolling. They just walk around, and we say, well, they're looking for something to do. Well, basically they're sampling stimuli, and then the appropriate stimulus will come in, and then they will engage in some kind of an act. If you look at different bees, and you look at the probability of responding to a certain set of stimuli, they're going to be very different. For instance, the bee on the top, this represents her profile over time, and it can change with time. So over time, hers is changing in this way, and this one on the bottom, and over time, hers is changing this way. So at any given time, they have different response thresholds to different stimuli. They're unique with respect to their profiles of what they're likely to respond to. This particular one, maybe when she went through and sampled the environment she patrolled, it might have been the right combination of stimuli exceeding thresholds that result in collecting pollen. And this one, perhaps nectar. And these are changing all the time. They're not fixed. No individual has a fixed set of response thresholds. They change with age. They change with learning, which is an exponential change. They change with development. Their prior experience, their genotypes set them differently. Lots of things lead to individuals having different profiles of the set of response thresholds that they use to sample the environment.